Chapter 19, Section 2, ATP Synthesis. All right, so let's talk what happens next. Previous section, we went through how all the complexes, complex 1, 3, 4, can take excited electrons from that have been chaperoned there by NADH, give you QH2. QH2 transfers those electrons via complex 3 to cytochrome C. Cytochrome C brings those excited electrons to complex 4. 4 bleeds off the last of the energy, transferring it to the final electron acceptor water, giving a metabolic of water, and in the process, 4, 4, 2, 10 hydrogens have been pumped. Well, what can you do now? Well, those hydrogens, high concentration here, low concentration here, p side, n side, intermembrane spaced matrix, depending upon you're talking mitochondrial, you're talking cytoplasmic membrane if it's a prokaryote. Well, those hydrogens are now going to power ATP synthase, our ATP synthesizing enzyme. ATP synthase ends in A, so it's an enzyme. What does it do? It's a synthesis enzyme, hence synthase. What does it synthesize? ATP. Remember, in science, if you don't know what a word is, break it down into its parts and work backwards. A's, synthase. Synthase of what? ATP. This drives the synthesis of ATP as the hydrogens literally flow back across the membrane. Remember, while they can diffuse across the membrane, that's going to be extremely slow. They flow, move very quickly back across the membrane through this enzyme, this protein, ATP synthase. As they're flowing back through, ATP synthase is going to take a diphosphate, inorganic phosphate, produce ATP and some water. So you have the hydrogens flowing from the positive side to the negative side, in a membrane to the matrix, outside the cell to the cytoplasm of the cell, if it's a prokaryote. What we see happens is you inhibit the electron transfer, you inhibit ATP synthesis. No respiratory chain, no ATP synthesis, because you shut down the electron transport chain. There's still going to be a hydrogen differential across. Those hydrogens are going to flow through until you go from extremely high to extremely low and everything balances out. And then ATP synthase is going to stop doing what it does. There are a couple of ways to uncouple. Uncoupling means you're going to get rid of this, transfer of electron energy, the pumping of the hydrogens. DNP, FCCP, these are weak acids that carry protons across the intermembrane space, so they will basically get rid of that differential. No hydrogens high on one side, low on the other side means that no flowing of the hydrogens through ATP synthase. So you can go through and you can remove any oxidizable substrate. So that means you get rid of the organic materials, this acetyl-CoA, any of the succinates, any of that. You get rid of that, but as long as the hydrogen imbalance is there, ATP synthase will still continue to occur. And they've shown it. You have to have some protons that are able to move across. And as long as they can, things will continue to work. The problem is once that imbalance balances out without anything there to oxidize, thus giving you the electrons you need, this will shut down in time. Someone just touched my foot, freaked me out. So this is what they refer, this ATP synthase is what they refer to as an F-type ATPase. Has two functional domains, F0 and F1. Now remember, this is just the mitochondrial ATP synthase. If you're talking about a prokaryotic one that's at the cytoplasmic membrane, it will be different. It is similar in structure and mechanism, but the ones you're going to find in bacteria and in chloroplasts will be slightly different, because remember, there's a couple of billion years separating them. So there will have been differences over that time. The two domains, F0, F1. F0 is the integral membrane protein with a proton pore. Okay, so this is what the protons, the hydrogens, will flow through. The F1, oh, this is a peripheral membrane protein. Remember, peripheral, it's not actually in the membrane, it's associated with the membrane. This is actually what catalyzes the hydrolysis of ATP, the making of ATP from the A diphosphate in the inorganic phosphate. F1, F0. F1 is where the actual enzymatic, quote-unquote enzymatic activity occurs. F0, well, look at this. This is a structural. It's embedded in the membrane, and it's holding the F1 components, the alphas and the betas, in position. Okay, when you look at this, ADP plus inorganic phosphate to make ATP and water. Okay, it is technically a reversible reaction, but the thing is, the interactivity of the F1 catalytic units to ATP is tighter for ATP than it is to ADP. It goes back to what we were talking about earlier. All of these reactions, while reversible, left to right, right to left, if you draw it out the chemical equation. But by having tighter bond, looser bonds, this, that, or electronaught values that are E0 values that are way different, you can tilt the reaction more one way than the other. Well, here the reaction's tilted left to right, ADP to A triphosphate, ATP, because of the interaction with the F1 subunits to ATP, not so much to ADP. So it's tilting the reaction more left to right, less right to left. Greatly reduced right to left. So not only is the proton motive force driving this reaction, it is also driving the release. Typical enzyme, okay? Remember this, E plus S, you get the intermediate ES, and then the release of P. E, the enzyme plus substrate, you get the interaction of the enzyme substrate complex formed, and then the product release, the product being at a higher energy state than the original substrate. Well, with ATP synthase, ADP plus phosphate, enzyme ADP, enzyme ATP, ATP released. So you notice it goes down, down, and then you get this really big hump in the release there. This, overcoming this activation energy state here needed, that's where the hydrogens flowing through come into play. The F1 subunits, 3 alpha, 3 beta, gamma, delta, and a sigma. Each of the beta units is one catalytic site for ATP synthesis. The betas change shape. Beta ATP, beta ADP, beta empty. Beta, 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 ADP, ATP, empty. Notice the ribbon models for the three states of beta show different structures. F0, F0 has the composition of A, B2, C, N, N depending upon the species. So I could go and I could isolate mitochondria, isolate the ATP synthase from those mitochondria, and then you hand them over to you and you could take them to a lab and you could sit there and tell me which group it comes from. Maybe you can't tell me exact genus species, but you can tell me, oh, this is an animal, or this is of this derived animal, this is more mammals, insect, whatnot, based on the number of C rings, which is composed of the C subunits. B, A, C. Here's the artist rendition of what we were just talking about. A, B2, and then however many rings, 8 to 17, which pass through the membrane. A is where the hydrogens flow through, causing everything to spin. Here's the alphas, the betas, the gammas, the epsilon of the F1. Here's the the F1 subunits that will take ADP, ATP for the betas, and then will produce. Going from intermembrane space to the matrix is where the hydrogens are flowing through. This is the computer ribbon model of everything that the artist has drawn here. The C rings are going to have proton binding sites so that as the protons flow through the A, they're interacting with the rings, causing the rings to spin, which causes gamma to spin. Gamma spins causes the subunits, the catalytic subunits, to also spin. They refer to this as rotational catalysis. Okay, it's the flow of the protons through the F0, specifically the A, interacting with the A and the rings, causes the F1 to change conformation, to also move. We see the betas will go through three states, three conformational changes. Tight, loose, very loose. If it's ATP, it's tight bound. If it's ADP, it's loosely bound. If it's empty, it's very loose binding capacity. So the betas, A diphosphate, ATP, empty, and it'll just go through. ATP is released. It's now in an empty state. It'll loosely bind ADP, becomes ATP tightly bound, released, and it just keeps going through. And you got to remember, this is also spinning, moving, because F0's got a conformational rotation going on due to the flow of the hydrogens, which is causing also these to move. So as these are spinning, they're changing their state on loose, very loose, loose, tightly bound. And what we're looking at here is the flowing of hydrogen. So you have kinetic energy, which is causing the spinning, the rotation of F0 in the gamma subunit of F1 to now spin. So now the kinetic to mechanical. The mechanical energy of, which is leading to the changes in states interacting with the beta subunits. And you have the creation of, or the synthesis of, ATP. don't want to say creation because we don't like using the word creation in biology or sciences. We say synthesis. So they've studied this. They've isolated the ATP synthase. They have attached it to actin filaments and then had hydrogens flow across, and they've studied the conformational change, the movement of the different components. So what they think is happening is you have the membrane and you have the catalytic subunit, or you have the units here, the C rings of F0. And you have electrons, or the hydrogens, as they're moving through, are causing this to change its position so that they actually will now start moving, spinning. So you get a transient protonation of glutamic acid here, glutamate, in the subunits, which causes this to change position, to move. So you have the hydrogens flowing up and in, and then this one, because it's now protonated, will move. The next one comes into place, flows in, and then moves. As they get to a certain place, they interact with other proteins, and then that hydrogen will be released. So this moves, causing the gamma subunit of F1 that's interacting with this to also now start to rotate. How many proteins, how many rings, and all of this is going to be different, species to species. How they're oriented. Here they're showing 13. Well, this one here is showing 14. Different species, different numbers. High, low numbers, doesn't matter. They're still going to operate the same way. The overall equation for ATP synthesis, so many ATP plus so many inorganic phosphates plus O2, hydrogens, NADH, gives you ATP, water, metabolic water, and NAD. It's kind of one way to look at it. This taken and not taken into account where the electrons are coming from or whatnot. So this is a very basic look at what's happening with ATP synthesis. You'll see the textbooks, some biochemists will discuss the PO ratio, or the P2E, to electron ratio. Gives you the ratio of how many electrons are needed for the production of ATP. So they figure for every ATP, about 2.5 electrons have to enter in at the complex 1, or 1.5 at ubiquinone. Remember, because you got those certain things where FADH2 or whatnot can just directly go to ubiquinone. They can bypass complex 1, complex 2. So, less energy, is lost. More hydrogens can be pumped because, you're bypassing that second law of thermodynamics by reducing the number of transfers. This ratio varies, species to species. We see that one of the things it's going to be based on is how many subunits in the F0 complex. We also see that this proton motive force is what brings the ADP in to the mitochondria. Because remember, a lot of this ATP is being used throughout the cell. Okay, this isn't like the chloroplast where any ATPs are just going to be used there, you know, that are generated via photosynthesis is going to be used there for the Calvin cycle. No, the ATPs generated here in a mitochondria are used everywhere in the cell. So, somehow, they need to be moved out. They need to be, you know, that ATP has to be taken out of the cell. And then once it's been cleaved, hydrolyzed from ATP to ADP, those ADPs have to get back in. Well, turns out we have an adenine nucleotide translocase, an antiporter. Remember, antiporters are transporters that move things in different directions. ADP in, ATP out. Proton motive force is what's powering this. It's what's getting this occur. ADP in, which the F1 subunit of ATP synthase will turn into ATP. Well, where's the inorganic phosphate coming from? Well, right there. H2PO4, another, is with a symporter. Symporter things that transports things both in the same direction. Hydrogen's flowing through here, just like it's flowing through the ATP synthase. But as it's flowing through here, it's moving, you know, the phosphate in. So now, here's your inorganic phosphate when this gets cleaved. Here's our ADP. Here's our synthase that's going to join the two up. They call this a synthosome. ATP synthase may be making the ATP, but without this antiporter and this symporter, there's nothing for it to work on. So they always have to be working together, hence a synthosome, just like we had the replosome. Got to get those NADHs from the cytoplasm, from glycolysis into the mitochondria. So you're going to have shuttle systems. Malate aspartate shuttle, glycerol 3-phosphate shuttle. When you look at what's happening here, NADH, what they call NADH equivalents. Yeah, the NADH that's actually made by glycolysis, the NADs that are reduced to NADHs, don't actually travel into the mitochondria. No, that's what they mean by this equivalence. Notice here, oxaloacetate that's in the cytoplasm will be reduced by malate dehydrogenase. Difference between oxaloacetate and malate, carbonyl hydroxyl. Well, guess what? We've already talked about the fact that there is a transporter that can take malate from the cytoplasm and make it malate and just move it into the mitochondria. One side of the membrane, other. Well, then you just use malate dehydrogenase in the mitochondrial matrix to oxidize malate back to oxaloacetate. There's your NADH. Here's the NADH you started with. The NAD stays out. NAD that's inside now gets reduced to NADH, and then that goes off to our respiratory chain. And, you know, okay, malate to oxaloacetate. Well, guess what? You can't have that oxaloacetate outside there. So guess what? You're going to take oxaloacetate and you're going to convert it to aspartate using glutamate and alpha-ketoglutarate. Hey, guess what? The difference between oxaloacetate and aspartate is this amino group. Where are you going to get the amino group? From glutamate. Turn glutamate into alpha-ketoglutarate by, stripping off the amino. Transfer the amino to aspartate. Well, there's a glutamate aspartate transporter. Boom. Out it goes. Aspartate gets converted back to oxaloacetate because there's a glutamate alpha-ketoglutarate here. Alpha-ketoglutarate, which was out in the mitochondria, gets transported out. Here's our malate alpha-ketoglutarate transporter. It's an antiporter, malate in, alpha-ketoglutarate out. Our glutamate aspartate transporter is also an anti-transporter, an antiporter. Aspartate out, glutamate in. So notice, aspartate, oxaloacetate, malate, malate, oxaloacetate, aspartate. It's just a cycle. And in the process of this cycle, NADH to NAD, NAD to NADH. Outside, inside, inside, outside. Boom. You get something similar with the glycerol 3-phosphate shuttle. So this is outside the mitochondria, or outside the cell, or in an intermembrane space, whatever. Glycolysis gives you NADH, NADH to NAD, because you're taking dihydroxyacetone phosphate, turned into glycerol 3-phosphate. Boom. Glycerol 3-phosphate. Backs to dihydroxyacetone phosphate because you're stripping off carbonyl, becomes hydroxyl, FAD becomes FADH2, which those two hydrogens are passed to Q. Q goes to complex 3. Everybody's happy, happy.